Well, good morning. Thanks for getting up early and coming to hear what we have to say. So from a standpoint of fisheries, I have kind of a passion for this. I grew up on Martha's Vineyard, and there was really nothing else to do but fish and dive. And I come from a family that has a long history in commercial fishing. My grandfather's a commercial fisherman on the vineyard for 65 years, and my mom's family had been on the vineyard for about 200 years. So it was thought that I was either going to be a carpenter or a commercial fisherman, and I crossed over to the dark side when I actually was the first to go to college and actually get a degree in marine biology. I have still some family members that don't like to talk to me because they think I'm the enemy. But the important thing is that when I started my studies in marine biology, I realized that fisheries were something that I was truly interested in and passionate about, because one of the things is I love to fish. And I want to see people to be able to continue to fish, but that's getting more and more difficult because of the state of our oceans. So one of the things that I've focused a lot of my research on, and great working with Kelly, because Kelly can help me answer some questions that I couldn't answer myself, as Dr. Livingston was pointing out. Okay, so the bottom line is, for the last 40 or 50 years, people recognize that seafood is good for you. It's healthy for you. It's a great source of protein that's low in fat. It's loaded with omega-3 fatty acids. It's good for your heart. It lowers the risk of obesity, heart disease, stroke, all these things. So we've been told in the last 40 or 50 years that seafood is good to eat. And you know what? People have listened. But not only that, the great thing about seafood is it comes in all different varieties, tastes, flavors. So the great thing is there's something for everybody in seafood. So not only is it healthy for you, there's something for everybody. And it all comes down to, the bottom line is, we have a love for fish, a genuine love for fish. This person, as you can see, has a greater than normal love for fish. But the bottom line is, over the last 100 years, the technology that we use to go out and harvest the fish that we love has greatly expanded. And not only that, there are now billions of people on the planet, all of whom love fish. So what this has meant is that we're now seeing significant increased pressure on many of our live stocks of fish. And remember, it was only 200 years ago, 150 years ago, that marine biologists said it was absolutely impossible to fish out a marine fish species. 150 years ago, the most prominent marine scientists in the world said it was impossible. And we're there. We're there. So the bottom line is human populations have increased dramatically over the last 50 years. We can see human population growth, but we also see that the number of landings of fish have now increased. We're now landing somewhere in the neighborhood of over 7 million tons of fish per year. So with this increased human population growth and increased demand, because seafood is good for us, we are now starting to see problems in our oceans. And many of the populations are the things that we like to eat. In fact, right now, fish harvest is increasing at a rate of 13% per year. That's unbelievable. So the thing is that originally people fished to live. We fished for subsistence. That's how we survived. And then, interestingly enough, there are some people who got really good at fishing, but they were horrible farmers. So what happened is this led to the evolution of commercial fishing. You could trade your fish for corn if you were really good at catching fish. So what happened was we see a shift from subsistence fishing towards more commercial. And over the last 100 years, commercial fishing has exploded largely based on technology. We have ships that can span the ocean. We have ships that don't have to come into port. They can fish 365 days a year because they have fuel ships that come out to supply them. They can or freeze fish right on board. They offload it. They're constantly out at sea. So as a result, the technology has enabled us to keep boats fishing all the time. But what we're beginning to see, particularly in the state of California, is a loss of our commercial fisheries. Now what we're seeing is a shift from more commercial to recreational fishing. And in Southern California, with 22 million people, you'll be shocked at how many people sport fish in Southern California. So trends in world fisheries. Marine fisheries now are landing about 70 million tons of fish per year. It's estimated that the first sale value of that fish brings in over $50 billion worldwide. So we're talking major, major contributions to worldwide economy. Fish dominate the landings. They account for 80% of everything that's landed in terms of seafood. So we're talking fish here, not necessarily crabs or lobsters or all those other mollusks that we like to eat. And of the 17,000 species of fish found in the marine environment, believe it or not, 50% of that 7 million tons comes from only 20 species. That's it. 20 species constitute 50% of that. So presently, there are few environments of the ocean, that is vast on our planet, that are not currently being exploited for some marine resource for food. So fishing technology used to be limited by the technology that we had. How fast your boat was. Could you refrigerate or prepare fish at sea? Now that's no longer a limitation. The only limitation that we have are the economic costs of doing that. Because when it comes to you going to the supermarket and buying the fish, once fish gets over $20, $30 a pound, you're not going to buy it anymore, which means it becomes too expensive for somebody to go catch. So we've also seen a shift in fishing pressure from what used to be, it was the commercial fishermen that were responsible for the decimation of fishing in our oceans, to now, with all the people we have on the planet, we're saying increased significant pressures by recreational fishers, something that people never thought could happen before. So the bottom line is, it's hard to just point our finger at the commercial fisheries and say they've been the source of the problem. Recreational fishing has become very popular, and after World War I and World War II, there was actually federal initiatives to encourage people coming back from the war to go fishing. It was a great way for them to reunite with their families, to get back to the things that were inherent in being an American. So in Southern California, it's estimated there are over 1 million sport fishers in Southern California alone. They generate over $1 billion in revenue to the economy from sport fishing alone. So you can't tell people that they can't sport fish anymore because it's actually part of our culture. For many of us, our most fondest memories come from fishing with our fathers or our grandfathers. So now we're seeing a state where our fisheries are being depleted, our resources are being depleted. Commercial fisheries are going out of business because they can no longer be viable. They've overcapitalized in many cases. Recreational fishers are being told they can't fish anymore in their favorite spots because we've depleted many of these resources. But these have important effects on not just our economy, but our culture. So, California fisheries are not immune. We've seen this not just worldwide, but in California as well. So we've had 100 years of heavy exploitation here in California. And here we just have a really basic graph. On this axis we have fish size. These are the size frequencies of the various fish. So this is from 1984 to, oh, we're losing juice, Kelly. There we go. To 1995, and what you see is that fish are getting smaller and smaller and smaller. That red line that you see is the size at sexual maturity, which means now for rockfish, this graph is for rockfish, most of the rockfish that are being landed in the fishery haven't reached sexual maturity yet. They haven't had a chance to spawn, which means their chances of replenishing the population are going down. This, for a fisheries biologist, is not a good sign. In addition, we've lost a lot of the critical habitat that many of these species used in the last 100 years. We've filled in many of our wetlands. We've filled in many of our estuaries to build marinas, to build condos. And as a result, these are critical habitats for many of these species. So these species are getting a double whammy. We're not only taking the adults out of the population, but we're taking away critical nursery habitat for their larvae. We have pollution, both chemical, thermal, siltation, as the result of coastal development. We have shifts from commercial to more recreational impacts, which changes the way managers go about managing these fisheries. So this, remember, just sport fishing alone contributes $1 billion to our local economy. So it's not so simple to come in and say, we have to protect fish, we're going to tell everybody you can't take them anymore. That is not a viable solution. One of the things we've been focusing on at Cal State Long Beach in our fisheries research is providing solutions that keep people fishing but help mitigate for some of these problems. So, I'm going to talk a little bit about the live fish fishery in California. This fishery evolved to supply primarily Asian seafood markets, both in California and worldwide, with fish. And the way it works is you go into the fish market and you actually pick out the fish that you want. They'll take it out and they'll cook it for you right there on the spot. So this fishery evolved in California in the late 80s. It peaked in the 1997, and it targets primarily rockfish, flatfish, cabezon, and sheephead. And generally what they're looking for is a fish big enough to fit on a plate. So when they cook the fish for you, they cook the whole fish. It's beautifully displayed, but the fish has to be big enough to fit on a plate. So as the result, for every single one of these species, because of the demand, we saw a significant decline in catch rates for all these species. And when we see that sign of decline, that's usually a bad sign. Now, to give you an idea how much these fish are worth, the average value of live fish is $18 per pound. The typical fish landed ranges between 2 and 4 pounds, so you're talking $36 to $72 per fish. That is a valuable resource. So as you can imagine, this fishery was hammering these populations pretty hard because it was very lucrative for fishers. So one of the key species we want to look at that differs from all the rest is this California sheephead. So the California sheephead is a local species. Woo, those colors. I'm colorblind. Does that look rough to you? Okay, sorry about the font. They range from Baja to Monterey, California, but a heart of their populations are found in Southern California. They're found at depths of about 150 meters, which is about 300 feet. They're typically found over rocky reefs and kelp beds. They eat things like sea urchins, crabs, brittle stars, amphipods, eating primarily the little invertebrates that live on the reef. Males can reach 3 feet in length. So most of these, only the biggest individuals will get that big. Most of them stay around 12 inches or so. So there are 2 fisheries for this species in California. One is the commercial sport fishery, which targets primarily using hook and line or spearfishers, and they target primarily the big, large males. And as a result of depletion of the stocks, California State Fish and Game implemented a minimum size limit of 12 inches in 1999. But there's also a commercial fishery, this is the live fish fishery, that targets the species using both traps and what's called stick gear, it's hook and line gear. And they're generally targeting the females, which are plate size, small enough to fit on a plate. So they also implemented a minimum size limit in 1999 at 12 inches, and then they raised that to 13 inches in 2001. So the state was trying to mitigate for the population decline. So if we take a look at this graph, you can see that the commercial landings, total commercial landings, were actually very low up until the 80s, and recreational fishing of sheephead was the most prominent part of the fishery. But starting in the late 80s, when the live fish fishery took off, commercial fishing exceeded the sport fishing landings. And these 2 fisheries combined were landing over 370,000 pounds of sheephead a year. Problem was, we didn't really know anything about the biology of this species. So Fish and Game was scrambling to get a handle on how to manage this when we don't thoroughly understand its biology. So one of the ways they go about doing this is managers will implement bag limits. A fisher can only take X number of fish per day per angler. Or they'll set quotas for the commercial fishery. They can only take so many tons, and then the fishery gets closed for that year. They'll implement restricted access, which means only a certain number of commercial boats can actually fish in the fishery. Or you'll have season closures. You can say, oh, you can only take them outside their breeding season. And then, of course, there are size limits. So the bottom line is all of these harvest control techniques require a catch and release, which was something that we were very interested in. So the way size limits work is you have small females. Those small females reach a size where they reach sexual maturity. So that minimum size at sexual maturity, you want to allow all the fish in the fishery to have at least 1 season to spawn. So if you're going to set a minimum size limit, you want to set that just beyond that minimum size. That'll allow all the individuals in the population at least 1 season to spawn before they can be harvested, which means any of the fish below that size you have to throw back. But all the fish bigger than that size you can keep. Question is, and the problem is, Fish and Game was implementing these minimum size limits, and they did not know whether the fish that were caught by anglers and thrown back actually survived. So fishers are going, wait a minute, you're going to make me throw back fish that could die? Why can't I just keep those? That doesn't seem right. So we figured this would be a very fruitful area of research. So catch and release is based on this concept of survival. So if you force fishers to throw back fish, some of those fish may not live because of the impacts of actually catching them, bringing them up on the boat, measuring them and saying, oh, they're too small, throwing them back. But some of them may survive. Fisheries managers go, that's good, but there can be problems. Just because they survive doesn't mean there aren't sublethal effects. There could be physiological effects that could impair growth, that could affect their reproduction, that can impair their immune function. So even though they live, they could be set back as the result of this practice. And in addition, these physiological impacts can influence their behavior. And these behavioral things can influence how fast they move, how much they eat, all sorts of factors. These are things that we don't know before Fish and Game is implementing these regulations. They didn't know the answer to these questions. So what we want to know is, what are the effects of catch and release on sheephead? Fishers have to throw them back, but do they even live? Do they survive that process? So we wanted to quantify their behavioral stress that comes from being caught either in a trap or on a hook and line. We wanted to quantify their physiological stress, and I'm a pseudo-physiologist. I have to collaborate with folks like Kelly, who's a real physiologist, or Kevin Kelly, who's our fish endocrinologist. I think he'll be talking at the next Fellows Breakfast. So I enlist their help to help me with the physiology, but I was working on the behavior, and I got some help for them to do the physiology. And we wanted to compare both the commercial practice of catching fish in traps and the recreational practice of catching them on hook and line. If you catch them, if you throw them back, are there impacts? So, and the question was to determine how many survive. Fish and Game didn't even know that before they implemented these regs. So to measure the behavioral stress, what we do and what my lab specializes in is we put acoustic transmitters on fish or in them, spy technology I call it. We can let the fish go and we can follow them around and see where they go. So of course the problem is when you catch a fish and you surgically implant these tags, you affect its behavior. So we needed a control for that. So the control that we use for that is sometimes we hide the bait, we hide a transmitter in a piece of bait and then we feed it to them. We don't have to touch them. Fish doesn't know it has a transmitter, and we can follow the fish around and we can compare the behavior that we see from fish-fed transmitters to those that we handled and then surgically implant transmitters. And then we can measure things like how fast they move, how much area they use to determine the impacts of catch and release. And then using Kevin Kelly's tools, we can take a blood sample from these fish and we can look at things like some of their endocrine responses or biochemical assays to determine their physiological stress as a result of catch and release. So one of the things that we can do is we can rapidly catch a fish, take a blood sample, and before the stress hormones begin to circulate, we can determine its pre-stress level. Then we can hold fish for 10 to 15 minutes. Those hormones begin to circulate, then we can get a measure on how stressful that practice is. So what we did was we went out and we caught fish on hook and line, and we used the same methods that most anglers use. And then we'd pull the fish up and take a blood sample, we'd take a measurement, put a transmitter in the fish, hold it for 15 minutes, take another blood sample, and let the fish go. Some of the fish we actually caught in commercial traps, just like commercial fishermen use. We'd bring those up and sample the exact same way. So we're comparing both recreational and commercial type fishing. So the fish were brought up, we would anesthetize them, we'd do surgery. Many of my students have been taught how to do surgery. We can make an incision in the body wall. We can insert these transmitters, which cost about $300 a piece, so they're not cheap. We'd stitch the fish up, flow the fish over, put an external tag in its back so we could identify it if we saw it again underwater, and then release the fish. And then the students could then follow around and track the fish for periods of up to 72 hours. In addition, while the fish was out, we could take a small blood sample from the caudal vein. This is like going to the phlebotomist and getting a little blood sample. We would take a blood sample as soon as we brought the fish up, and then after the surgery, we'd take another blood sample and hold that fish, then we'd release them. So here's a fish that we would done surgery on. Here you can see the tag, the fish is swimming around. And then we can even follow fish underwater using underwater hydrophones. So quite often, not only could we track the fish from the boat, we could follow them around underwater and make sure they looked okay. So the bottom line is the tracking part is very laborious. I have a student here that's doing this now. She spends a lot of time in this boat. So the bottom line is you put the transmitter in the fish is underwater, the transmitter produces an acoustic signal that can be detected by a hydrophone. Somebody has to follow the fish around, but by doing that, we can know the exact location of the fish over a period of 24 to 72 hours. So here is a location over at Catalina where we did all this work, and this is an area called Big Fisherman's Cove, and it's a marine protected area. So this red dotted line you see is part of the marine protected area. Each one of these polygons that you see represents a neighborhood of a California sheephead that we tracked. So the first thing that this indicated to us was these were fish that we tracked over periods up to a month. We noticed that all the fish that we caught surgically implanted transmitters in all survived. No problem with survival. Even though we used the same practices that normal fishers use, plus we did surgery on those fish. So these represent the home ranges. And the other thing I want to point out is that most of these fish spend most of their time inside this little marine protected area. So of the 24 fish that we tracked, somebody out in the boat following them around, we detected all the fish, and the average time of the battery length of the transmitter is about 40 days. We detected all fish for at least 34 days. We also used long-term acoustic transmitters that last a year. We had underwater receivers that are constantly listening for the fish in the area. We detected up to 16 fish for 318 days. That was the life of the transmitter. So we were able to confirm basically 100% survival, regardless of the fishery method that we used, and regardless of the fact that we actually surgically implanted transmitters. Fishers don't do that. We wouldn't encourage fishers to do that. So despite we do all these things to these fish, we know that they're surviving. So when we look at their behavior, we see some interesting trends. So remember, these are the fish that we caught on hook and line. These are fish that we caught in traps. So these fish had transmitters surgically implanted into them. But those fish over there are fish that we fed transmitters. We didn't touch them. So the first thing that we noticed, if we look at their rates of movement, how fast they're moving over the course of the day, fish that were line caught and trap caught move significantly slower than those that we just fed transmitters to. So this indicated to us that there are some behavioral impacts of this catch and release and the surgical implantation. If we compare that with how much area or space they use, we notice that fish that we caught and handled used less space. They not only swam slower, but they used less area. Now, as you can imagine, when you get out of surgery, you're not going to be running marathons, right? You're going to take a day or two off, and you're going to be moving a little slower. So we do know that there are behavioral impacts as a result of this. However, if we take the fish that we did surgeries on, and we look at their first 6 hours of tracking, we see that they're moving significantly slower than 48 hours later. So within 48 hours after surgical implantation, these fish are moving at the same rate as the fish that we fed the transmitter to. So within 48 hours, those behavioral effects of catch, release, and surgery are gone. So if we compare that to Kevin Kelly's physiology, we see some really interesting trends. So one of the things that Dr. Kelly was looking at was, and my axis labels have disappeared. On this axis, we have cortisol. So everybody knows that when you get stressed out, cortisol is a stress hormone that enables your body to deal with the stress. If you want to experience stress, come here during finals. You'll see students coughing and sneezing and experiencing all sorts of immune depression as a result of cortisol spikes. So what Kevin's students did was we took these fish, we brought them up, and that fishing practice stresses the fish. We take a blood sample, we hold them for periods of up to a month in tanks. This is what most people do when they measure physiological stress. What do we see? Cortisol spikes, and it remains elevated for prolonged periods of time. It actually takes, even after a month, they're still showing signs of chronic stress. If we compare that to lactate, so remember, we're catching fish on hook and line, they're fighting against the line, they're struggling. Remember, just like going out and running really hard, you build up lactic acid in your muscles. We see a spike in lactate, but that spike in lactate is gone within 12 hours, even in captive holding. Now, here's the interesting part. If we take fish that we capture, remember we held these fish in captivity so we could serially sample them again and again, look at their cortisol levels. They're stressed out. So we know that not only is this contributed by the process of catch and release, but some of it's being stressed out by being held in a tank. Here's that same curve. We took some fish that we caught, took a blood sample, stressed them, and then let them go. Then we went out fishing again and again and again, tried to recapture those fish. We didn't keep them. We didn't hold them. We let them go. Here are the fish that we recaptured over time. So this is 18 hours after we captured and stressed a fish. Remember, according to our data, this is where that fish should have been within 15 minutes after holding. So what this means is within 18 hours after catch and release, physiologically, cortisol is back to baseline. And it remains there. These are all the fish we caught and released. This has never been done before. Every study that's looked at stress in fish have kept fish in captivity. We now know that causes chronic stress. You can't use that as a sign of recovery. So this is the first study that's actually demonstrated both behavioral and physiological recovery from a catch and release event. So our conclusions, first of all, this catch and release practice implemented by Fish and Game works because we know, even despite the fact that we were doing surgery on these fish, 100% survival. In addition, there's both a behavioral and physiological impact of catch and release, but it appears to be fairly short-lived. Within 18 hours, fish are back to normal, both behaviorally and physiologically. Angling has slightly higher stress levels than trap, but not significantly, and behavioral and physiological recovery within 12 to 24 hours. So seasons, size limits, quotas, things that require fishers to throw back undersized, immature fish actually work. It should help the population recover. So with that, I'm going to turn things over to Kelly, who's going to talk about the other half of the story.
I have to apologize for my voice. I'm really sorry. Before I start, is this better if I have a microphone on, or does it make any difference? Keep the microphone on, okay. I normally have, like I said, a very melodious voice, so I'm sorry. Chris has told you quite a bit about the California sheephead. He's told you where they live. He's shown you some of their home ranges. And he's told you a little bit about the fishing pressure on these fish. But what he has graciously left for me to do is to tell you what I find the most compelling part about California sheephead, and that is their reproductive biology. Sheephead are sexually dimorphic. That just means males look different from females. So here we have a typical female. She's got a salmon color throughout with a white chin, and a typical male with a salmon color in the middle with a black head and a black tail. Males also develop a more prominent forehead. We call it a nuchal hump that they develop as they mature as males. So they look different. These fish are also haremic, and this means, just like it sounds, the males maintain a harem of females. Depending on the social status and the size of the male, he may maintain a harem of 1 female, 2, 4, maybe even 15. He has sexual access to those females, but he also has to defend them from other males that may be in the population. Finally, sheephead are protogynous sequential hermaphrodites. I know it's early, but yes, I said hermaphrodites. Typically, as humans, when we hear the word hermaphrodite, we think, oh, genetic malfunction. And it's true. Hermaphrodites, of course, where you have both testes and ovaries in the same individual. That can happen in humans. It's extremely rare, and those individuals are sterile. They're not fertile. This is a result of genetic malfunction. But in fishes, this is not the case. In fact, there's about 21 families in fishes, including the wrasse family, of which sheephead are a part, where hermaphrodism is quite common. Hermaphrodites in fishes can be either simultaneous or sequential. Simultaneous hermaphrodites have both ovaries and testes working in that one individual at the same time. So they're producing eggs, they're producing sperm at the same time. Sequential hermaphrodites are all born one gender and then transition or sequence into the next gender at some course of their lifespan, or they can do this. If the hermaphrodites are sequential, they can either be protandrous or protogynous. Protandrous or proto-andros, so first male. They're all born male, and then at some point in their life, they can transition into female. That means those testes turn into ovaries. The most typical example, or the most common example of this that I've seen at least, are the anemone fish or the clownfish. So it's true, the rumors you've heard, Nemo was a hermaphrodite. We can also have protogeny, so protogyne, so first female. This means that the fish are all born female in a population, and at some point, that ovary that was producing estrogen, that was producing eggs, transitions and becomes a sperm and testosterone producing testis. A great example of this are the sheephead. So here you see this gorgeous male who, just to clarify this one, that used to be a female, right? He was born a female and at some point transitioned. This is why I find the sheephead so interesting, how they can switch their sex like this. So you now know what this phrase means technically, protogynous sequential hermaphrodites, right? But let's think about this for a minute from the point of view of managing the fishery. Chris told you that the fishery, the population levels are going down. To manage this fishery, sorry about my voice. To manage this fishery, usually you use models with a male and a female, so you have two genders. But remember, here in sheephead, we have males, we have females, and now we have a third gender, an intersex fish that we call transitional fish. This throws a lot of those computer models out the window. You can't manage this in a traditional way because now you have this additional gender to deal with. Now, if you were managing a fishery, you might also be interested in what induces a female to transition to a male. What would cause this? In fact, this has been very well studied. It's not age, it's not size. In fact, it's changes in social structure, changes in the population structure. Here we have our harem. So we have a large male, he's got a large harem of females. If that male were to die or to be removed from the population, then the most dominant female would transition into a male. So that same individual would become a male. So here she's got her harem. Her ovaries become testes. Her female sex behavior becomes male sex behavior. She becomes a male. Now, sheephead have been doing this for centuries. This is what sheephead do. Right? This has been going on with no problem. But now we have to have this added lens when we look at this population change through the lens of fishing pressure. Now, Chris told you about the two different types of fishing, sport fishery and the commercial fishery. So, and their different targets of large males or plate-sized females. So if we look at this harem again in an area with a lot of recreational pressure, where they're targeting those large males, and you would see large males being removed from the population at a very rapid rate during the fishing season, what you might find happening as this continues to occur, that the females might not have time to gain the size and the body mass that they would have before transitioning. And you might see changes in the size of males at transition. Because of this idea that fishing pressure may actually be changing the population structure of sheephead, in addition to the fact that the sheephead fishery management is based on data collected in the 1970s. Excellent data, but data collected in the 1970s. And as Chris showed you, fishing pressure has changed substantially since then. This really led us to this collaboration to understand what does the population look like now? What's out there right now? So our objective, our objective was multifold. First of all, we wanted to get an updated report on the sheephead population. We wanted to know how many males are out there, how many females, how many transitionals. Because again, if we have increased fishing pressure, perhaps we have an increase in this transition rate. We wanted to compare this to this historic study in the 1970s to see if anything has changed with fishing pressure. As a reproductive biologist, I wanted to know if they were functional during transition. What is that intersex gonad doing? And finally, we wanted to know about the different populations across the California coast. The historic study was done at Catalina Island. But of course, as Chris told you, the range of sheephead, at least in California, spans the whole California coast up to the northern Channel Islands. So we got, at the time, our undergraduate researchers out there, and they set out to quantify the number of males, females, and transitionals. And our initial part of this study was to start at Santa Catalina Island, right off our coast. So here we are, right? So right off our coast, because that's where the historic studies were done. The first thing that our students came back with us after they started catching fish was a little bit of a roadblock. You see, it turns out that yes, sheephead are sexually dimorphic, but just like humans who are sexually dimorphic, there's a range. So it's not that textbook perfect female versus male. How many males do you think are here? 3. One male. Oh, they're all male. Turns out that there's 2 males, 3 females, and 3 transitionals. So you see, our students actually became quite expert at judging this, but it's very difficult. Of the 116 individuals that we caught during that first year, they sexed 59 of them correctly, just based on this external morphology alone. And that's a percentage rate of 50%. Any CSULB student here will tell you that's not where you want to be, right? That's not good. So how are we able to know what they really were? Well, we went to the gonads. This is not a sheephead, but this is a cross-section of a fish, so you can see the gonad in situ, what it looks like in the body here. Here's an ovary. Gonads were removed, and then sections were taken from the gonad. They were embedded in wax, and the students sectioned the gonads serially into 6-micron sections. This took thousands of hours. They did this for hundreds of fish to do this, quite a laborious process. Once they were able, they took the slices and they put them on slides so we could look at what the gonad looked like under the microscope. And their results were beautiful. They did an excellent job. It's clear the difference between an ovary and a testis. That's for sure. So here we have an ovary, and here we have a typical testis. In the ovary, you can see these pale circles here. These are the oocytes that will eventually combine with sperm to produce a new individual. Those oocytes are surrounded by supporting cells, and I've circled the whole structure in blue. It's called a follicle, so our ovaries are full of follicles. In testes, you have developing spermatocytes here, some less mature and more mature. And this is typical of a testis, of a fish testis, where they develop in packets. The transitional gonads looked substantially different. This is at the same magnification, so the first thing you can see is that they're quite atrophied. We do see follicles, we do see packets of the developing spermatocytes, but nothing is mature. So our students answered that one of those first questions not right away, it took them a long time, but one of the first pieces of data that we got was that the transitionals are non-functional. They're not ovulating and they're not releasing sperm, which is important. Well, it may have been important. You might think, well, is this something that matters to have the transitionals non-functional? You might even expect that. In fact, if you look at the historic data, the data taken in the 1970s, you might not think that that's a big deal at all. Because the historic data at Catalina Island showed no transitionals during the breeding season. Sheephead have a breeding season, they have a non-breeding season. When you're in breeding season and you are in the wild, you must seize the day, right? You have to take that chance and you have to breed. This is it. This is your one shot to pass your genes to the next generation. You don't transition. You don't have non-functional gonads. You need to breed. So when would you transition? You would transition in the non-breeding season, and that's what they found. Warner found in the 1970s. However, our CSULB undergraduates found at Catalina Island, when they collected in 2004 through 2006, 26% transitionals in the breeding season. These data are from the breeding season only. This is percent of the catch, immature fish, females, transitionals, and males. The females, transitionals, and males are all mature fish. 26% of this catch is non-functional for breeding season, and that's a big deal if we're trying to manage this fishery. That's a really big deal. What we wanted to do then is to see, well, Catalina has a lot of sport fishing pressure. It's easy access to Los Angeles, to Long Beach. So maybe Catalina was unique. So we wanted to expand our study throughout the whole California coast to the Northern Channel Islands with different, less reduced fishing pressure or different anthropomorphic pressures. But before we could do that, we really needed to get a handle on the sexing of these fish. Because taking the fish, killing them, taking the gonads, going through serial section, that would be an enormous task. And also, besides that, we have students who would do it. But we wanted something that would be applicable, something that we could take to the California Department of Fish and Game and say, here's a quick test. Here's something you could do. Not, well, you could take out the gonads and spend thousands of hours sectioning them. No, they don't want that. So the first thing that we needed to do was to develop an easier, less invasive, less expensive method for sexing these sheephead. And this is what our undergraduate student, Carrie Loke, started, and now she's a graduate student in our program. She came up with a very complex series of measurements that she took on the fish. Maybe we couldn't tell at first glance, okay, black head, black tail, maybe that didn't work for sexing, but maybe there'd be a measurement like forehead depth, because remember, they developed that nuchal hump, or something else that if we just had a digital image of the fish would be easy to tell if it was male, female, or transitional. Carrie spent a lot of time doing this, digitally analyzing hundreds of fish. And she learned something that is true of science, that it doesn't always work. In fact, she found no difference for any measurement. And I'm showing you 5. She actually did 15 different measurements. Nothing worked. So this is standard length along the bottom. We have head area, head outline, forehead length, forehead depth, body depth. She had tons of measurements. Nothing worked. What these graphs are showing you are symbols for males, females, and transitionals all piled on top of each other. Nothing stood out. So Carrie set out to develop an easier, less expensive, less invasive method of sexing. Again, this time she went to the endocrine side of things. We had blood samples, like Chris showed you, from all of those fish, and you saw that blood samples could be withdrawn. You don't have to kill the fish. You could catch and release, and this would be a great thing for Department of Fish and Game. She set out to measure estrogen, which is made in high amounts by ovaries, and testosterone, which is made in high amounts by testes. And here she scored. So this is a graph of 17-beta estradiol. 17-beta estradiol is simply the most dominant estrogen in fishes and in humans and most mammals. So we have 17-beta estradiol in picograms per mil in the blood. You can see that immature transitional males have very low estrogen, but females have high amounts. And you would expect this. For testosterone, she measured 11 ketotestosterone, the most dominant testosterone in fishes, not in humans, but in fishes. And we see immatures have a nominal amount, females have none, or virtually none. Transitionals and males both have testosterone. So this is a good start. We can tell females she would have high estrogen, no testosterone. But we can't tell the difference between transitionals and males at this point with our endocrine assays. However, if we think about this reproductively, this does give us an insight into this transition of the gonad. Number one, it means that those males or those females, once they commit to transitioning, shut down that production of estrogen and immediately crank out testosterone. It must be an early response. So we're actually looking more into that study, seeing how soon that this testosterone begins as a possible clue to help us understand the transition. Carrie and Mike and a number of undergraduates in our laboratories have shown that transitionals are non-functional. During that breeding season, they're there at Catalina, and this is a big deal. They found that morphology, what the animals look like, is not necessarily the best way to sex them, but that we can use hormone analysis to provide alternative non-lethal methods for sampling the populations. Carrie has gone on. She's actually sampled sheephead from San Diego to the Northern Channel Islands. She'll be defending her thesis in a couple of months. I invite you all to come and listen to her data. She's done a fabulous job. And we've been able to report our findings to the Department of Fish and Game, letting them know about these transitionals in the breeding season, letting them know that the population out there is not what it appears, and that we've gotten great feedback from them. And so this is exciting that our research has been so applicable and will continue to be applicable. Now, I am out of time because we started late. I have 4 minutes or 3 minutes of what you can do as a consumer to pick the best eco-friendly fish. If you'd like me to do that, I can, or I can stop. Okay, all right, let's go. So usually when I start talking about, because I'm not a fish person, even though I guess this sort of doesn't say that. But I start talking about fish, gonads, and people they say, okay, yeah, you always talk about gonads, but tell me, what can I do? Do I buy sheephead or not? Do I eat sheephead or not? That's what they want to know. So that's why I wanted to end with something that you guys could take home, a little bit of information about what you can do as a consumer. So I have a few slides, some shopping tips. On buying fish. Some of this is from Sunset Magazine. I'll have that website up along with a number of other websites that you can use for information. So number one, the thing to do is make it Western. California, Oregon, Washington, and even British Columbia, we have the most restrictive, the most eco-friendly fishing policies of anywhere in the world. So look to see where your fish come from. It's better here than Atlantic. It's better here than in other countries. Make it Western, and you're making a good eco choice. Eat a variety of fish. I like salmon. You may like salmon. A lot of people like salmon, but if we only eat salmon, that's not good for the salmon. So there's a number of fish out there that are tasty, delicious, and heart healthy. And when you buy them in season, they're cheaper, they're fresher, and it's better for the fish. It's better for the environment. Finally, I get asked this question a lot, wild versus farmed. And the answer is, it depends. Salmon, wild Alaska salmon. I'm imprinting that on your brain. Wild Alaska salmon. That's the best. That's the most eco-friendly thing you can do. Or wild Pacific salmon, at least. Don't buy farmed, because it's not eco-friendly. But farmed rainbow trout, the rainbow trout fishery is actually very eco-friendly. So you have to kind of look in, and some of these websites I'll give you at the end will give you more information. A lot of people are concerned about toxins, mercury levels. You've heard this in your fish, mercury levels in the fish. Well, a very quick primer on bioaccumulation. Mercury is ubiquitous. It's everywhere in our rivers, streams, oceans. It's represented by the red dots in this diagram. If you're a small individual, you'll take up small amounts of mercury. If you're a large individual, you'll also take up mercury from your environment. But also, if you eat other individuals that have taken up mercury, you get their mercury load as well. So the farther up you go on the food chain, you notice that the mercury levels increase. Your fish shopping tip, don't buy top predators. Top predators have more mercury loads. Don't buy shark. Don't buy swordfish. It has the most toxins. Stay lower on the food chain if you can, like sardines. Or if you want to eat a top predator like salmon, go for something like salmon that's short-lived. Salmon mature at 3, 4, 5 years, as opposed to something like tilefish, who has 10 or 15 years to accumulate mercury. So this is a way to reduce the toxins that you get. Finally, it depends on how you cook it. Toxins are stored in the fat in fish. If you grill, the fat drips off, right? It goes away. If you deep fry the fish, then the fat seals in the toxins. It also, I think, cancels out the heart healthiness if you deep fry. So, okay, so no fish and chips. I'm ruining your whole morning by telling you there are plenty of fish left to eat. There's plenty of wonderful choices out there, beautiful fish that are healthy, that are safe, and that you can eat eco-consciously, without too much guilt. So, just a few of the choices. If you forgot what I just scrolled really quickly for you, you can go to Monterey Bay Aquarium. They have a little printout. It's not this large. It's actually pocket-sized. So you could take it with you. And they have best choices for seafood. It says best choices, good alternatives, and things to avoid. They also have best choices in sushi. Speaking of sushi, if you like to go out and you don't cook, the Long Beach Aquarium has actually partnered with a number of restaurants in Long Beach that promise that they will only use sustainable fish. And that's a big deal. So you can spend your money as a consumer and actually make a good environmental choice. So here are the websites that I promised you. I will leave these up here. The Seafood for the Future, this is from the Aquarium of the Pacific. And their website, Monterey Bay Aquarium, if you want to download those little cards to take with you to the store. The EDF.org will give you the mercury PCB loads, and it's a very user-friendly website. And finally, I got all those beautiful pictures from sunset.com and their shopping tips. So I want to thank you for being here early, for listening to my horrible voice. I'm so sorry. I want to thank our funding agency. Chris and I have a grant through California Sea Grant. We've had a grant through Department of Fish and Game, PSCO. The SCTC and the LA Rod and Reel are groups of fishermen in the community that support our students, and that is a huge deal. We really thank them. There's been about 1 million students working on this project. They've all done a great job, and we appreciate them, and I appreciate your time today.